Studies of Neanderthal DNA have shown that we are genetically distinct from Neanderthals. But the overall lesson of molecular studies of human evolution has tended to run in the opposite direction, revealing just how astonishingly close we are genetically to the rest of the natural world. In fact, molecular data have often challenged and overthrown long-held assumptions about human origins. The great chemist Linus Pauling was the father of modern molecular approaches to evolution. During the early 1960s, he and Emil Zuckerkandl compared the amino acid sequences of corresponding proteins from several species. These were the early days of protein sequencing, and their data were inevitably limited. Nevertheless, the pair noticed a striking pattern. The more closely related two species are in evolutionary terms, the more similar are the sequences of their corresponding proteins. For example, comparing one of the protein chains of hemoglobin molecules, Pauling and Zuckerkandl noted that over its total length of 141 amino acids, there is only one difference between the human version and the chimpanzee. But the difference between humans and horses is 18 amino acids. The molecular sequence data reflect the fact that horses have been evolutionarily separated from humans longer than chimpanzees. Unearthing evolutionary history buried in biological molecules is now common practice. At the time, however, the idea was novel and controversial. Molecular approaches to studying evolution depend on the correlation of two variables: the length of time two species or populations have been separated, and the extent of molecular divergence between them. The logic of this molecular clock is simple. To illustrate it, let us imagine some matchmaking between two pairs of identical twins, one of genetically identical females and one of identical males. Each female is wed to one of the males, and each couple is then placed on its own otherwise uninhabited island. From a genetic perspective, the populations of the two islands are, at the outset, indistinguishable. Now leave each couple and its descendants alone for a few million years. At the end of this period, mutations will have occurred in the population on one island that will not have occurred in the population on the other, and vice versa. Because mutations occur at a low rate and because individual genomes, being large, offer huge numbers of possible sites where mutations might occur, it is inconceivable that both populations will have acquired the same set of mutations. So, when we sequence DNA from the descendants of each couple, we will find that many differences between the once identical genomes have accumulated. We say that the populations have diverged genetically. The longer they've been separated, the more divergent they will be. But how do we tell time, so to speak, by looking at this molecular clock? Put another way, how can we measure the genetic divergence between ourselves, say, and the rest of the natural world? In the late 60s, long before the advent of DNA sequencing, Alan Wilson, a whimsical New Zealander at UC Berkeley, together with his colleague Vince Zarich, set about applying the Pauling-Zuckerkandl logic to humans and their closest relatives. But at a time when protein sequencing was still a dauntingly cumbersome and laborious affair, Wilson and Zarich found an ingenious shortcut. The strength of an immune reaction to a foreign protein reflects how foreign the protein is. If it is relatively similar to the body's own protein, then the immune reaction is relatively weak. But if it's very different, the reaction is proportionately stronger. Wilson and Zarich compared reaction strengths by taking a protein from one species and measuring the immune responses it triggered in others. This gave them an index of the molecular divergence between two species. But to introduce a time dimension to this molecular clock, they needed to calibrate it. Fossil evidence implied that New and Old World monkeys, The two major groups of monkeys separated from their common ancestor around 30 million years ago. And so Wilson and Sarich set the immunological distance between New and Old World monkeys as equivalent to 30 million years separation. Where did this put humans in relation to their closest evolutionary kin, chimpanzees and gorillas? In 1967, Wilson and Sarich published their estimate that the human lineage had separated from that of the great apes about 5 million years ago. Their claim provoked an uproar. In paleoanthropological circles, conventional wisdom held that the divergence had occurred around 25 million years ago. Between humans and apes, the establishment insisted there is clearly much more than 5 million years' worth of difference. It was for many cause enough to dismiss the Berkeley team's newfangled genetic method as untrustworthy, and to declare that, anyway, geneticists should stick to their fruit flies and leave humans to the anthropologists. Wilson and Sarich, however, weathered the storm, and subsequent research has shown that their dating of the human great ape split was remarkably accurate. When the time came to extend his analysis of the human-ape divide from proteins to DNA, Wilson entrusted the effort to his graduate student, Mary Claire King. The product, in 1975, was one of the outstanding scientific papers of the 20th century. For a long time, though, such a triumphant outcome seemed unlikely, especially from King's perspective. Her work had not been going well, owing in part to the enormous distraction created at Berkeley by the anti-Vietnam War movement in the early 1970s. King considered going off to Washington, D.C. to work for Ralph Nader, but fortunately she sought Wilson's advice. If everyone whose experiments failed stopped doing science, he wisely counseled, there wouldn't be any science. King stuck with it. King and Wilson's comparison of the chimpanzee and human genomes combined a number of methods, including a clever technique called DNA hybridization. When two complementary strands of DNA come together to form a double helix, they can be separated by heating the sample to 95 degrees centigrade, a phenomenon called melting in the molecular geneticist's jargon. What happens when the two strands are not perfectly complementary, when there are mutations in one of them? It turns out that two such strands will melt apart at a temperature lower than 95 degrees centigrade. How much lower will depend upon the degree of difference between the two strands. The greater the difference, the less the heat required to pry them apart. King and Wilson used this principle to compare human and chimpanzee